So thank you very much for the invitation. I'm very grateful to be here today. And I would like to take advantage of this opportunity to start a conversation about the way we screen DNA sequences for biological risks. Let's start with the basics and what is biosecurity. So if you Google biosecurity, you will end up with a number of links to pages describing policies to protect agriculture from various diseases. And the bigger brigade, is in charge of preventing pests from entering the country at the airports. We've all seen them. So fundamentally, biosecurity is about preventing a biological threat from entering a community. Well, that was at least what biosecurity was until 2005. In 2005, scientists at the CDC used chemical DNA synthesis to resurrect the strain of influenza that was responsible for the 1918 Spanish flu pandemic. This pandemic killed over 35 million people worldwide in less than 2 years. So it was the most devastating pandemic since the Black Death. And that strain of influenza was not present on Earth anymore. There were some relics of those sequences in corpses that were buried in the permafrost in Alaska. And so people could sequence it and reconstruct the virus. And obviously, this was a very controversial research project because what if, even though there was a legitimate reason to do it, there was also a risk of a leak in the environment or people using the sequence for, I was told that people don't see my sequences. I apologize for that. Can you see my screen? Can somebody see?
No, we can't.
Oh.
John, would you like me to show your slides?
Give me another chance.
Okay.
Do you see a screen now?
No, we don't.
And now?
Yes, we're getting started,
Okay.
but not the slides yet.
No, you don't see the slide. Okay, sorry. Apologize. Hold on.
Okay.
You see the slide?
Yes.
Okay. So I apologize for that. So I lost my momentum. Yeah, so they were able to, that was somewhat of a controversial project that represented a certain biological risk. And that got people attention. So quickly after that, there was a English newspaper that published a story explaining how easy it was to get DNA fragments containing, a small part of the smallpox virus genome. And so now we are in a situation where our understanding of biosecurity is changing because, the threat doesn't need to come from the outside. It can come essentially from inside, essentially through a computer network, just through access to information. And the threat may not necessarily look like something that is outside of our community. And so this kind of attention was not good for the gene synthesis industry. That industry was just, pretty much starting at the time. So they very rapidly organized themselves and developed a protocol to screen their orders to detect the presence of suspicious sequences. Today, most of the gene synthesis companies and the number of biotech companies that are not within the gene synthesis business, interestingly, are members of the International Gene Synthesis Consortium. And you can go on their website where they publish their harmonized screening protocol. At the same time, the government also paid attention and they developed a document describing their recommendation about the screening of synthetic DNA orders. A draft was published in 2009, and a year later, the final guidance was published in 2010. It includes multiple sections such as customer screening, record retention, relation with the government, but the sequence screening was the one that we particularly pay attention to. And this document is actually fairly similar to the one that was proposed by the industry. So that's good. The goal of the sequence screening is essentially to detect sequences that may be embedded in gene synthesis orders that are specific of select agents and toxins. So select agents and toxins, that's a list of infectious agents that have been put together by the government that are particularly dangerous. And so it includes both DNA sequences, but also protein sequences. And they're looking for sequences that are only found in select agents and can be as small as 200 base pair. So essentially the screen goes like this, it performs a 6-frame translation of the DNA sequence. The DNA sequence is broken down into smaller fragments of 200 base pair. Amino acids, the proteins are broken down into short blocks of 66 amino acid sequence. And then you use, sequence alignment tools to compare those fragments to gene bank entries, and you look at the keywords associated with the annotations of the base hits. Then you analyze these keywords to potentially flag a sequence. So during the summer of 2010, I put together a team of students to do what I think was the first implementation of this guidance, actually, and to evaluate its performance, because it was not totally obvious to me what kind of results we would have when we implemented that screen just from the text that was in the Federal Register. So the good news is that on one hand, it worked, it's able to detect small fragments, the computational cost was acceptable, and it was relatively easy to implement, didn't require, an extensive curation effort of sequences. But on the other hand, there was a lot of interpretation that was necessary to do the implementation. There was no target accuracy for the screen. There was no way of knowing whether an implementation met the government expectations or not. And then we found also some major vulnerabilities in the way the screen was designed. So at the end of the day, you end up in a situation where different implementations of the same guidance could lead to completely different screening outcomes. And that's probably a little bit of a problem. We published that paper, those results in Nature Biotechnology, released the software open source. The students were part of iGEM, so they went to iGEM. At the time, security at iGEM was probably not the most interesting projects that you could, present. So the reception was a little bit lukewarm. I think that it has gone better, especially, due to the outreach that the FBI has done over there. So, I think that the iGEM community is much more aware of security now than they were 10 years ago. But the interesting part was that the day after the iGEM Jamboree, the students were invited to present their results to the government. And so they gave a 2-hour lectures to 30 people representing, I think, something like, 15 different government agencies. And there was much more interest from the government than actually from the iGEM crowd about those results. So the experience of implementing the guidance and seeing the reaction that we get after the work was published makes me think that maybe we've not been thinking about this in the right way. I think that there is a number of problems with the way the screen is performed today. First, the focus is way too narrow in terms of both what are the sequences that we are screening and what kind of threats we are looking for. Also, somehow the gene synthesis industry has been tasked with performing those screens, but it's not their job. Their job is to make DNA, not to do biosecurity screens. And for them, it's just a cost that eats on their bottom line. So they have actually a financial incentive to limit that expense as much as possible. Also, the malicious use of gene synthesis seems a fairly hypothetical threat at this time. And there are a lot of people who have been asked to do that, who essentially tell you, why would I spend so much effort into detecting something that I will never see in my lifetime? So I think that there are a number of problems with the way we've been doing it, and there is a renewed interest for this. And so maybe it's a way, it's time to be changing slightly the way we're thinking about it and broadening our perspective. So one way of broadening the perspective is to broaden the scope of what we are looking for. And in other areas of security, combination of things that are not dangerous on their own can be strong signals that something bad is about to happen. And so it really doesn't take a whole lot of imagination to come up with dangerous combination of DNA sequences. And I would invite you to, to do that exercise, spend 15 minutes, think about it. And I bet that you will come up with half a dozen strategies that would not require major equipment or special skills. And that may be very, unfortunate combination of sequences that are not dangerous on their own. The other category of sequences that we should be concerned about are the ones that don't match any natural sequences. And this is a plasmid that was designed in my group. And the sequence in blue, it's about 3 kilobases of DNA that was generated by a computer software. And when you run that against gene banks, you don't get a single hit. And I think that, this sequence would go through the current screening protocols very smoothly because it doesn't match any select agents. But I think we should be a little bit more concerned about 3 kilobases of DNA sequence that we know nothing about, that 200 base pair of any viral genome. Finally, I think that we need to put the screen in context. Some sequences that are not dangerous in the hands of qualified scientists could become dangerous in the hands of the, in the wrong hands. And like, for instance, if you take adenovirus, which is used in a number of clinical applications, it's safe, it's not on the select agent list. And that sequence coming from a pharma company would not raise any questions because, they make a lot of products that use that. However, the same sequence coming from a community biolab would probably deserve a little bit more investigation because, do-it-yourself gene therapy or genome editing projects is probably something that we should be aware of and want to reverse in some way. So in a previous life, I used to do a lot of professional flying. And when you fly an airplane, maintaining situational awareness is key to safety. And situational awareness is actually a model that can be applied to a number of different security and safety scenarios. And essentially, it says that decisions should be taken based on a number of environmental signals that are integrated to get a picture of the current situation and try to interpret where the current situation is going to lead you in the future. So we could apply this model to sequence screening. The signals that we could get are more than just synthetic gene orders. It's just one signal. Design files on the design files that are on our computers is another signals, our oligo orders, or even the DNA sequences that are produced by our DNA sequencers are other signals. The comprehension is really about the bioinformatics analysis of those signals to produce some information that is understandable by a human operator without a PhD in bioinformatics. I think that the projection now consists in connecting the results with people associated with these signals. No sequence is dangerous on its own. It's only dangerous in the hands of someone. And so the question is, are the sequences aligned with the activities that the people have disclosed or been approved to do? And if there is a discrepancy between the sequences that they are working with and what we think that they are doing, then this is a red flag that requires some investigation and some action. And the action is not necessarily, oh, you shouldn't be doing it. The action may be, well, we need to update the scope of the approvals or the, of what you've been doing because, we miss some information, or maybe you want to rethink that project or that order and do something else with your time and your way. So it seems that the purpose of sequence screening doesn't need to be limited to looking for a bad guy trying to use gene synthesis for malicious intent. And it's actually fairly well aligned with the standard biosafety policies and practices that we already try to abide with. And we actually have a great biosafety system in this country. I don't think that biosafety incidents need to be reported. And I spent some time trying to find statistics about biosafety incidents. I didn't find any except, anecdotal evidence that the track record is actually pretty good. Number of laboratory-acquired infections is very small and things like this. And it's somewhat similar to the outstanding safety record of the air transportation system. And I think that when you put the two side by side, there are striking similarities in the way they are organized. except for one thing, that the biosafety world doesn't have any technology. We don't have any software. We don't have any biosafety technology system that really makes it possible to implement a lot of the policies and the training and things like this. But I don't think there is any reason it could not be developed. And I would be very interested in talking to anyone who might be interested in trying to figure out what this may look like. So I'd like to acknowledge the students who were part of the 2010 iGEM team. It's interesting that a lot of them did very well and have become data scientists over the years. And I would also like to acknowledge the people who sponsored that project.